Hello, my name is Emma Smith and today I will present human immunity. In today's presentation, I'm going to cover the following topics. I'll begin with a summary of what pathogens are, then I will follow with a breakdown and explanation of the first and second lines of defence, both of which are non-specific. I will follow this with a discussion of the third line of defence, going into detail about acquired immunity, sensitivity to antigens, as well as the roles of different types of immune cells and immunoglobulins. After this, I will explain what is meant by non-self and self and why this is important in regard to transplantation. I will then discuss two immune system disorders, specifically Crohn's and multiple sclerosis. In the last section of this presentation, I will describe the categories of acquired immunity, the immune system response to vaccination, including immunological memory, the benefits of vaccination with an example of how it has affected measles infection rates, and I will then discuss the concept of herd immunity, vaccination programs, and factors affecting vaccine uptake. So firstly is pathogens. So pathogens are organisms that cause disease. They are communicable, which means they can be spread between people. Pathogens enter the body through orifices, injuries, the respiratory or digestive system. There are different types of pathogen. Firstly, we have parasites. These live in or on hosts, can be single or multi-celled. They can be worms or mosquitoes, for example. Next, we have protozoa. These live in moist habitats and are single-celled organisms. Then we have fungi. These typically cause skin or bloodstream diseases. They are spore-producing and can be single or multi-celled. Then we have prokaryotes. These are also known as bacteria and are single-celled and cause a wide variety of diseases, from leprosy to cellulitis and UTIs. A virus, this is the first of the non-living, it is not alive until it finds a host. Examples of viruses include HIV and COVID-19. A prion is also a non-living pathogen. It's a type of protein that triggers normally healthy proteins in the brain to fold abnormally. So first of all we have the first line of defence. This is non-specific in that it does not distinguish between one type of pathogen or another. It will respond in the same way to all pathogens. It is present from birth and effective against a wide range of pathogens. The first line of defence can be broken down into two categories, physical and chemical barriers. Physical barriers prevent pathogens from entering the body. The skin is perhaps the most effective of the physical barriers as it covers the entire body, it is waterproof and is made up of several robust layers. As you can see in the diagram here, we've got the epidermis, the dermis, and the subcutaneous tissue. Other physical barriers include mucous membranes, which line the surface of the eyes, throat, nose, and gastrointestinal tract, for example. We have peristalsis, which is the contractions within the digestive system, and physical expulsion, such as sneezing, coughing, and passing urine. Chemical barriers include secretions within the mucous membranes. These secretions include enzymes such as lysosome, which break down and destroy pathogens. In addition, within the gastrointestinal tract are stomach and intestinal juices, which are highly acidic and create an environment in which pathogens cannot survive. There's also vaginolactobacilli, which is a bacteria that metabolizes glycogen and forms lactic acid. This inactivates potentially pathogenic bacteria and viruses. Next is the second line of defence. The second line of defence is also non-specific and known as the innate immune system. It is predominantly considered to be inflammation, which is the immune system's response to damage or infection. Symptoms of inflammation include red coloration, warm to touch, swelling and pain. The purpose of inflammation is to increase blood flow to the area so that immune cells can gather and attack the pathogen. The process of inflammation begins with injury, which is caused by physical or chemical means and the pathogen entering the body. This causes chemical signals to be released by active macrophages. Macrophages are a type of white blood cell that kills microorganisms, removes dead cells, and stimulates action of other immune cells. Following this, mast cells at the injury site release histamine, which causes nearby capillaries to widen and become leaky, thus increasing blood flow and allowing for the passage of further immune cells, specifically leukocytes. Chemokines are released, which then also attracts phagocytic cells. Phagocytosis is explained in more detail on the next slide. But once phagocytosis has occurred, then the site and tissues are cleared, resulting in the healing of the area. So here we have phagocytosis. This is the destruction of defective, damaged, foreign and invasive cells. Phagocytic cells include macrophages, neutrophils and leukocytes. They target the cells that need eliminating and attract them with their receptors. Once they have interacted with the pathogen, they envelop and engulf it. They then digest the pathogen, breaking it down into proteins and other molecules, linking it with a lysosome within the cell. Once digested, the pathogen is destroyed. Now we have the third line of defence. Before we begin the third line of defence, though, it's important to discuss antigens. Antigens are found on pathogens and are the component which tells the body it is being attacked. An immune response is created by the body, demonstrating sensitivity towards antigens. This encounter with antigens causes the immune system to create antibodies, which in turn destroy the pathogen. The immune system will remember the antigen after its first contact, so that if it ever comes across it again, it knows which antibodies to produce and can make them at rapid pace. This is called immunological memory and is acquired immunity, which, in a nutshell, is the body's third line of defence. So, immunity can be acquired naturally, actively, or artificially. Natural immunity is acquired in utero, where the mother's antibodies are passed to the fetus via the placenta. This continues through breast milk once the baby is born. Active immunity is acquired through exposure to pathogens. This is where the body's natural immune system naturally responds to pathogens, creates antibodies, and develops immunological memory. So if the body encounters the same pathogen or antigen again, it knows which antibodies it needs to create to fight it. Finally, immunity can be acquired artificially, which is either actively or passively. Active artificial immunity is vaccination, where the body is exposed to antigens minus the living infectious agent. Passive artificial immunity involves introducing pre-formed antibodies to the body to generate an immune response. Next, we have 5 different types of white blood cells. Acquired immunity is thanks to immune cells, in other words, the white blood cells. Monocytes are the largest type. They become macrophages when they exit the bloodstream. They carry out phagocytosis. They can digest large particles in a wound, for example. Eosinophils help with allergies and worm infestations by releasing toxins. If an individual has high eosinophil counts, they are likely to be having an allergic reaction. Basophils are the rarest type of white blood cell. They secrete anticoagulants and antibodies to tackle hypersensitivity reactions in the blood. They act immediately. They release histamine to help capillaries widen and increase blood flow to a site, thus bringing more cells to tackle the invasion. Lymphocytes are part of the humoral immune system, which is the pathway by which antibodies are produced by B lymphocytes, also known as B cells, to target exogenous antigens. Lymphocytes are present in lymphatic tissues such as the spleen, tonsils, and lymph nodes. There are 3 types of lymphocytes: B cells, T cells, and natural killer cells. T cells help recognize and remove infection-causing cells, and natural killer cells kill virus and cancer cells. Neutrophils are the most common type of white blood cell. They are first on scene to an infected area. They attach to the wall of blood vessels to block germs from entering the bloodstream. They are also phagocytes. Next, we have a breakdown of antibodies. Antibodies are known as immunoglobulins, and they are glycoproteins. Antibodies are produced by lymphocytes, which are B cells, to eliminate infections, toxins, and antigens. They are made of light and heavy chains, typically structured like the letter Y, although some options can vary in shape. They have antigen binding sites at the end of the Y. Antibodies attack antigens by binding to them. The binding of an antibody to a toxin, for example, can neutralize the poison simply by changing its chemical composition. In other situations, antibodies can render the antigen completely immobile, or they can cause a chemical chain reaction which can cause the microbe to burst, which then attracts phagocytes to engulf and destroy it. There are 5 types of immunoglobulin. They vary in abundance, can be found in different parts of the body, and respond to different invaders. Now we have non-self versus self. The immune system needs to be able to identify what is self and non-self. Anything that is self is safe, and anything that is not non-self should be attacked, and the immune system must respond. The immune system looks for markers known as MHC, which will be explored in more detail on the next slide. These will identify the cell or antigen as self or non-self, as friend or foe. These self-markers are called major histocompatibility complexes, MHC, and these are how the immune system identifies self and non-self. There are two types, MHC1 and MHC2. The following table outlines the features of both. MHC are produced by all cells except red blood cells, and they are found on the surface of all nucleated cells. A cell such as a phagocyte will degrade foreign proteins and break them down into fragments. And when these are combined with MHC and displayed on the cell plasma membrane, this will indicate if it is non-self or self. MHC2 is produced only by antigen-presenting cells. These include macrophages and B cells. These actively ingest exogenous antigens such as viruses, toxins, pollen, and bacteria. These antigen-presenting cells break down these antigens and combine them with MHC2. This process triggers them to be recognised as non-self. Now we have transplantation. Transplantation is when a person receives an organ or tissue from someone else. Even if it is essential for survival, the recipient's immune system may reject it because it has detected it as non-self. Rejection can be classed into three types: hyperacute, which happens within a few minutes of transplantation; acute rejection, which is 1 week to 3 months after surgery; and chronic rejection, which takes place over many years. Immunological rejection is either direct or indirect. Direct is where the MHC, the self and non-self markers, warn CD8 lymphocyte T cells that the cells within the transplanted organ, for example, are non-self. The body's response is then to destroy the cells. This results in inflammation, thrombosis, secretion of cytokines, recruitment of leukocytes, and graft ischemia. And indirect rejection is the same as direct, but it also includes CD4 cells, warning lymphocyte B cells. This results in them changing into plasma cells and proliferation of T cells, which triggers secretion of antibodies, which are flags for destruction, and consequently passing macrophages to attack. Now we have disorders. There are over 100 autoimmune disorders as demonstrated in the image. This is where the immune system attacks the body because it cannot distinguish it as self. It sees it as non-self and produces antibodies to attack its own tissues. It can be triggered by bacteria, drugs, environmental and chemical irritants, as well as genetics. Autoimmune diseases are not curable, but they can be managed with medicine, therapy, and surgery, for example. It's also important to lead a healthy lifestyle with a balanced diet, lots of rest, a good amount of exercise, vitamin supplements, and avoiding known triggers. I now will explore two specific autoimmune diseases, Crohn's and multiple sclerosis. Firstly, Crohn's is an autoimmune disease where the body attacks the tissues within the gastrointestinal tract. As shown in the diagram here, you can see a healthy gut versus a non-healthy gut. Crohn's disease is a serious form of irritable bowel syndrome which is lifelong and incurable. The main symptoms are diarrhea, stomach aches, blood in stools, fatigue, and weight loss. It can be managed with medicine, surgery, or a combination of both, but it cannot be cured. Next, we have multiple sclerosis. This is where the immune system attacks the brain and or spinal cord of the nervous system, which results in fatigue, difficulty walking, vision problems, numbness, tingling, stiffness, and spasms. The immune system attacks the layer that surrounds and protects the nerves called the myelin sheath. This damages and scars the sheath and potentially the underlying nerves, meaning that messages travelling along the nerves become slow or disrupted. It is incurable but can be managed with steroid medicine or disease-modifying therapies, for example. Now we have acquired immunity. So acquired immunity, as mentioned earlier in the presentation, is either actively or passively achieved. Active immunity is acquired either naturally from contact with antigens or artificially through vaccinations. Passively acquired immunity can be natural, for example, obtaining antibodies from the mother while in utero, or artificially, where an individual is injected with antibodies already made. The immune response to vaccination. Vaccination is artificial immunity. It contains a weakened or harmless version of a pathogen, either as harmless pathogen fragments, dead pathogens, or toxins produced by pathogens. Despite being harmless, it stimulates white blood cells to produce antibodies tailored to that pathogen. If that is ever encountered again by the body, it has an immunological memory now, and it can produce the antibodies required at rapid pace. Vaccines prime the body for subsequent exposure. There are two types of vaccine, live and inactivated. Live vaccines are derived from wild or disease-causing bacteria, which have been weakened in a lab. Examples include MMR, rotavirus, smallpox, and yellow fever. They are effective with one dose. Inactivated vaccines are produced by growing bacteria or virus, and examples include hepatitis A, flu, polio, and rabies. They require multiple doses to be effective. Now we have the benefits of vaccination. There are many benefits to vaccination. Some include providing lifelong protection against fatal or life-altering diseases. They provide protection where antibiotics cannot. Vaccines have eradicated certain diseases such as smallpox, and they've prevented millions of childhood deaths. They also ease pressure on the NHS because people do not need the treatment if they'd caught the disease, and they can also work against cancer, specifically tumour-associated antigens. Vaccines, by helping large swathes of the population, has created stronger and healthier generations, which has consequently encouraged strong economic growth. To demonstrate the success of vaccines and how they work at reducing disease, the graph here on the slide shows the number of deaths per year since 1940 in the UK attributed to measles. Prior to the introduction of the MMR and measles vaccine, the measles vaccines first being in 1968, MMR in 1988, the Department of Health recorded between 160 and 800,000 notifications of measles. However, following the introduction of the vaccines and achievement of coverage levels in excess of 90%, measles transmission was significantly reduced, as you can see on the line graph here. Now we have herd immunity. So herd immunity is where a high percentage of the population is vaccinated or immune against a certain disease. It makes it a lot harder for the infection to spread. This is because there are fewer germs around to infect others. And if a person does get sick, an outbreak is unlikely because so many people are vaccinated. Some people, generally the high risk and vulnerable, depend on herd immunity because they can't have the vaccine themselves. Some diseases can't be prevented through herd immunity though, for example tetanus, which is caught from bacteria in the environment. Herd immunity may also be less effective because of poor vaccination rates, as anti-vaxxers may have encouraged people not to get vaccinated, or the pathogen may simply evolve too quickly or have many variants. There's also different diseases have different thresholds for herd immunity. For example, measles requires at least 94% herd immunity to be effective, whereas polio is typically between 80 and 86% herd immunity to avoid an outbreak. The World Health Organization supported herd immunity through vaccination during the COVID-19 pandemic. By encouraging people to get vaccinated, it brought about herd immunity and reduced the risk of people catching it and becoming very ill or dying. Next, we have vaccine programmes. There are lots of vaccine programmes throughout the UK. Probably the most well known is that for newborns, which offers vaccinations at different stages for diseases such as diphtheria, meningitis, measles, mumps and rubella. There are also several vaccine programmes such as the flu jab, which happen seasonally. These go to groups who could fall seriously ill if they caught it, such as the elderly or pregnant. The most famous of recent times is the COVID-19 vaccination programme, which was rolled out at very quick succession to the entire UK population, focusing first of all on the most vulnerable. Reasons that might affect vaccine take-up include anti-vaxxers, which encourage people not to take up vaccines because they believe that there are serious side effects. However, none of these have actually been proven as correct. To summarise, today I've discussed many aspects of human immunity, beginning with pathogens, the 3 lines of defence, non-self and self. I then discussed immune system disorders, acquired immunity and immune response to vaccination. I concluded the presentation with a review of the benefits of vaccination, herd immunity and vaccination programmes. Human immunity is undoubtedly complex and is a finely tuned machine to help keep the body safe. However, it can become disrupted and start to attack the body instead. It is also helped significantly by vaccinations, which has had a huge impact on society today. Vaccinations help the body to fast-track its immune response to many fatal and dangerous diseases. Thank you for listening.